If you don't have a syllabus yet, I have quite a number here. Does everybody have a syllabus? If you don't, then pick it up at the end of the class, please. I'll leave them here out in the room. Okay, so welcome to cell biology. This is my 15th year of teaching cell biology, and it's the second year in this classroom. And they've updated the technology in the classroom, and I'm trying to work with it. Not all working as it should, but we'll get there. Okay, so my name is Marinus Pilon. I'm in the Department of Biology. My office is E416 on the 4th floor. There's my phone number and my email. There we go. And office hours I will do Thursday afternoon and Wednesday morning. So Thursday 3 to 5 and Wednesday 10 to noon. So Friday 10 to noon. I do it on the Thursday and the Friday because there are labs on the Monday, Tuesday and Wednesday. Okay, so don't hesitate to call if you have questions or if you need explanations or for whatever reason. This year we have a new textbook for this class. So that's this book by Carp. There it is. It's lighter than the book we used before. I think it's also cheaper. But the real reason that I went to a new textbook is I think this is a much better textbook than what we had before. I researched this extensively. I think this is the best textbook for you. Because I think it's readable, I think it's, well, concise, but still it covers the material pretty well in good depth. So I like that. So, textbook, you can also go for an electronic version. You have that option. You will need the textbook, I think. There's also a lab associated with this class, and the labs are once a week, not this week because we have MLK Day on Monday. So there are labs, there's one on Monday, and there are 4 sections on Tuesdays and 3 sections on Wednesday. And for the lab, you will need a lab manual. Lab manual, very nice color. Right side up, lab manual. You can get via the CSU bookstore. It's something like $15 or so. It's not that much. For the lab, you will also need some form of a hardcover notebook for your lab notebook. Those are great, and you have to hand that in every now and then to our TA. So you need a hardcover, something like this. Is that clear? Right. The lab coordinator is Christine Prince, and I'm also involved in managing the labs, but I'm more distantly involved. So Christine is doing the day-to-day management. So if you have some kind of an issue with the labs, then you have to contact Christine Prince. And her contact information will be on the lab syllabus, which will be available to you in the first week of labs. We have 4 TAs for the labs, 4 excellent TAs, I have to say, because I always ask for and always get the best TAs available. It is really true. Okay? So we have Alpana, she's very experienced, Andrew, Jennifer, and Margaret. It's really a terrific team, I think. And then we have 1 extra TA for the grading of the lecture exams, and that's Charlotte, and she's here. And Charlotte will have office hours also in the weeks after the exams are returned to you. So if you have an issue with the addition of your grade or an assignment of points in a certain question, you will first go to Charlotte. If you don't figure it out there, then you can always still come to me. Okie doke. So that's that. We have a website, and that's on Ramsey to Blackboard. You'll find the syllabus there. You'll find PDFs of the PowerPoint slides. You'll find reading assignments. I post study guides. You'll find something called tasks, and that's something special that this course has. So there will be a number of exams. On the exams you can earn points. Now, you should understand that I do never curve in this class, never have, and I've honestly never been in need to do that. But there is a way in which you can get still some extra points back, because I understand that you can make mistakes on exams, and sometimes I got, unfortunate mistakes, and maybe you just forgot to learn something, and so I think it's good if you can go back and learn it again. So for the first 2 exams, you'll get actually a chance to get the points back that you've lost. 40 points you can earn in total. But you don't always just get it automatically. What I mean to say is, you have to do a little bit of homework. And you have to demonstrate to me that you do that homework, and that is what the tasks are. So the tasks are a form of homework. Most of those are assignments that come through the web, and that you turn in via the web in the form of online quizzes. And it's all doable. So if you participate enough, you accumulate enough of these what I call task points, so that you are allowed to do your exam corrections. So the message is, do your task homework, and you can do exam corrections. The tasks are all designed for you to help understand the material better. That's what I hope. Okay, also on the website you'll find old practice exams. Not yet, but they'll be up there in a moment. A couple of days. You will find your grades. There are communication tools. For lab reports, they're handed in to the lab TAs, but we're using this Safe Assign program that ensures that you are all doing original work. So that's on there. And then, if everything is correct, then right now this lecture should be recorded, as should be any lecture here, and so you can look that up later on the web. I haven't seen it work yet, but they told me it should be working. So you should come to class, but you can still look at the web and see what's on there later. And this is something that I asked last year, the students in this class, what they thought that I could do to improve, and they thought that this would be a great improvement. We'll see. Okay, there are a couple of exams. The first exam is actually handed out to you this Friday. It should. And it's a take-home exam. It's worth 50 points. It's a refresher. You can do it at home. You can use the book. You can ask me questions. It's a multiple choice form. There are 50 questions. And it's basically a recap of what you should have learned in introductory biology and introductory chemistry classes. I also refer to some pages of the book that you can go to just to help you complete that exam. So that's the first one. Now there are 3 midterms, and the dates of the midterms are on the syllabus. The midterms are here in this classroom, and you should plan to take the exams all the time scheduled. There's one final. I think it's on the first day of the finals week, also in the syllabus. Okay, so how do we grade? The exams together, that is the take-home exam and the 3 midterms and the final exam, are together worth 750 points. The lab is worth 250 points, that adds up to 1,000. So the lab is 25%, the lecture is 75%. The exam corrections add back into your lecture grade. So the take-home exam is 50 points, the midterms are each 150 points, and the final is 250 points. So the final exam in this class weighs relatively heavily. Okay, that's the grading scale. So if you get If you have more than 1,000 points, you can ask, how is that possible? Well, actually, if you didn't lose many points on your first midterms, I'm still giving you a chance to earn the 40 points for the exam corrections, but then you do an alternative assignment, so you can get above 1,000. If you achieve that, you get an A+. Usually I have 2 to 5 people who achieve that in a class this size. Then if you're above 900, you get an A, above 800 up to 899, a B, on and on and on. Alright? Okay, so somewhat unfortunately, I believe, this class has a reputation of being quite difficult. And I think it is a somewhat challenging class, but you know, we shouldn't exaggerate. So I really think this class should be doable, and I'm doing everything I can to help you, to guide you, and actually to make the class as doable as we can. Still, you know, a lot of people consider this challenging, and maybe it is because you have to do some abstract thinking. Cells are small, you cannot see them, you have to imagine what's going on at that cellular level. It can be challenging. So some tips. The exams are about what I cover in the lecture, and added to that, very specifically indicated assignments. So there will be a very, very specifically indicated assignment. If it's not in the lecture, you usually do not have to know it, unless I really have told you. See maybe an example today or on Friday. I really recommend to attend the class and to make notes. I do post the PowerPoint PDFs, and you can use those to print them out, bring them to class, and write on for your notes, so you can make your own notes. I do believe that making your own notes really helps. Research has been done extensively into how students and you learn. And it turns out that actually putting the PDF files online does not help you. There's no evidence that it has an adverse effect, but there's certainly no evidence that it has a positive effect. The only thing that really seems to help student performance is if the students in the class take their own notes. So you write up. Why does that help? Because one, then you have to listen. So you have to be focused on the lecture and what's going on there, so that helps. And two, the act of writing something up makes you remember. And in addition, if you actually do it right and you rewrite your notes after the class, you process the material, then you have to understand. And understanding the material in context is really what we want you to be doing. We want you to see the material in context, and by taking notes, rewriting them yourself, it's the best way to study. And because I am such a firm believer in that, the very first task that I will give you in a couple of weeks actually involves writing a summary. I will let you summarize something, and one of the summaries will actually be the summary of a class lecture. Okay. So attend class, make your notes, read your notes in the same day, read the assignments that go with each lecture if there are assignments. And then that point here, make your own summaries and models. That is so important. It involves some use of time, up to 3 hours for every hour of class, I would say. But doing that makes you learn the material much more efficiently. Okay? So I recommend that you study from your own summaries that you've written yourself. That's, I think, the most efficient use of time. If things are unclear, go back to the book, use that then as a reference. If it's still unclear, just come in and ask me. There will be practice problems, there will be tasks, you should do those. Overall, in this class, 99% of the students do all the tasks, and it helps. Don't postpone studying up to right before the exam. That's not what you should be doing. And then my final message there is you might not really learn directly from me. You know, I'll give you a lot of information, but really the learning process is something that happens in you, in your brain. So you do it yourself. Now, I hope I can help you learn. You are learning yourself. Learning is an active process. And then, of course, do not hesitate to ask questions. Okay. Well, any questions so far? All right. Let me tell you a little bit about myself. Let me tell you a little bit about myself. I am originally from the Netherlands. That is the country of windmills and wooden shoes. I did my PhD in 1992 in the field of biology and biochemistry. And for my PhD, I studied protein transport in cells, specifically protein transport processes in plant chloroplasts. I then did a postdoc until 1998 in the lab of Randy Sheckman, who, by the way, last December got the Nobel Prize for medicine and physiology. I visited him just a couple of days ago, and I gave him a bottle of wine, like that. And asked for what it was like. So it's kind of interesting. Since 1998, I've been here at CSU, and I am teaching classes. I've taught classes in genetics, cell biology since 1999, I think. Also introductory biology, life 1 and 2 that is. And I also teach a graduate class in plant metabolism and biochemistry. And presently my research is on the assembly of the photosynthetic machinery in plants. So, that's me. And okay, let me see if I can figure out how to spin works. I think I can. So, hello. So, I've told you about me, so it would be nice if by the end of the class you write me something back on a piece of paper, maybe. You write on a piece of paper. Now let's say you give me your name. And you give me that by the end of the class, so I'll give you some extra time for that. So your name, let's say your major. Get to use paper, so good. Your major, and let's say your year. Ready. And I'm always interested why you take BZ 310. And let's say you tell me what you might think of doing in 5 to 10 years. So let's say your career goals. Terrible handwriting, right? Career goals, and just anything else. So anything else that you might think is interesting for me to know, like if you have any concerns, or just if you have anything funny or interesting to say about yourself, like you're raising parents or whatever. For instance, for me, my favorite TV shows are American Idol and Downton Abbey. Yes, I'm very average. All right. And I'm really looking forward to the sports year 2014. I'm a big fan of soccer and the World Cup and the Winter Olympics, and I hope will not be a mess. Being from the Netherlands, I'm of course rooting for the Dutch speed skaters. I'll tell you how they are doing. All right, so now, should we be able to yeah, it worked. What didn't work was This pen should be able to write on the board. I could a week ago, but the system tricks me. I'll do it. Okay, so let's start with some material. The first week or so, I plan to do some reviewing and just lay a foundation. Okay, so you should read chapter 1, I think, which I have highlighted here in red. Let's read chapter 1. And then within the first and second week, also look at chapter 2 and 3.1. 2 and 3.1 give you an introduction into basic chemistry. These are things that you should have learned already. And chapter 1 is really an introductory, nice chapter. I like it. Okay, so we're going to recap a little bit, and we'll look in the first periods of the class at the basic structure of cells, what components we find in there, and then what I call the central information processing system, which is really how cells go from DNA to making proteins. Okay, and then we'll follow with that. Okay. So first of all, cell biology is a science. So, and in science, there are different methods that can be used. And an important approach is what we call this approach that uses hypotheses. So call it a hypothetical deductive or scientific method. I don't like this word scientific method because there is not one scientific method that wouldn't do justice to all of science. So, but in cell biology, the use of hypotheses is pretty important. So that's what we're doing. So what do we have? We have an observation. For instance, I could have the observation that I have here a microphone system. And now let's just say the microphone is not working. Let's just say the microphone is not working. So, now it is, but let's say it wasn't. So if it wasn't working, then I can say, well, okay, maybe the batteries are dead. Or maybe the microphone there is just broken. So to test the hypothesis that the batteries are dead, what I can do is I can replace the batteries with fresh batteries. By the way, you will be seeing me do that experiment frequently. I know the battery often run out. Okay, there are two outcomes of that experiment. If I replace the batteries and the system then works, then I know the batteries were dead. Okay? So then I accept my hypothesis, batteries were dead. But if I do not fix the microphone by replacing the batteries, then I have to reject the hypothesis that it was the batteries, because I have new ones in there. Of course, I need to do controls, and my experiment needs to be reproducible. So in this case, the control could be that, hey, maybe I brought batteries, but the batteries were already dead. So if I take my batteries that I used to replace and I test them in some other piece of equipment, like I bring a flashlight, and I get the flashlight to work with my batteries, and my batteries were not dead. That's a control. Also, my approach should be reproducible. So I should be able to describe exactly what I'm doing, and if there's someone in Japan using the same microphone system, they should be able to use the same approach. And it's the same for cell science. We should describe our experiments, including the appropriate controls, in such a way that people elsewhere on the planet can reproduce our experiments. Okay, so my hypothesis was a tentative explanation for an observed phenomenon. Microphone doesn't work. Hypothesis, battery is dead. That's my tentative explanation. This is a testable explanation. Okay. I can also come up with the hypothesis that someone on Mars is sending special signals to the planet Earth, interfering with only my microphone. And these are undetectable rays to any of us. That is not a testable hypothesis. I cannot test that. I cannot do much with it. So hypothesis can be tested, and if we test it, the hypothesis can be eliminated, but it can never be fully proven. So if I replace my batteries and my microphone works, then I think that my batteries were a problem, but maybe there was a loose contact in the system, and just me fiddling around with the system, I moved something on the inside and the contact was there again. So, there's never definitive proof in science. It's important to understand that. So we get a result. From the result we draw a conclusion, and the conclusion allows us to tentatively accept our hypothesis, but it's a tentative acceptance because there can always be alternative explanations that we haven't tested, or to reject the hypothesis. But the best experiments really lead, or the best experimental approaches really use hypotheses with experiments that have a high probability of rejecting the hypothesis. This is kind of counterintuitive to students, but I do believe in that. So in my own research, in my own lab, I always encourage my students if they have a certain observation and they come with an idea, then I say, okay, what is the most rigorous experiment you can come up with that would allow you to test this idea? And the best would be if you come up with an experiment that would really totally destroy your hypothesis. And then if you do that test, and the hypothesis withstands that test, then you feel much better. So you want to test as rigorous as you can. Okay, enough about that. So a hypothesis is a tentative explanation, and it leads to a testable setup. That's a hypothesis. Now, if we have a lot of hypotheses together, a lot of observations together, you get into repetitively, you see the same over and over and over. Now, if I have this, the police card, the police was doing instruction with this one. You all know that if I let this card go, it will drop onto the table. There's nobody in the room who thinks it will not be the case. Very predictable. This is because this type of experiment is done over and over and over, and it's because of Newton's laws of gravity. So if we get really a lot of consistent evidence, we get to a law, a scientific law, that very precisely predicts what's going to happen. Now, we might also have a large body of evidence, several laws together. This may lead us to a theory, like the theory of evolution. The theory of evolution is based on a lot of diverse evidence, a lot of different approaches, and it encompasses a lot of different hypotheses. Okay? Now, important to understand is that anything in science, whether it is a hypothesis, a law, or a theory, it's all changeable, it's dynamic. It all depends on the evidence that is available. So, for instance, Newton's laws of mechanics are true under conditions that we normally see, so pretty low speeds and masses that aren't too high. But if the speed goes up, then we get deviations from that Newtonian physics because of observations that Einstein and others got into. So we get relativity. So that's actually a modification of what was originally a very firm law, Newton's laws of physics. Yeah? How are we doing? All right. So what I just told you is that science is dynamic, and that things are not so fixed. Nevertheless, I'll try to tell you things that are true. And apparently, some professors have said in the past. I'll let you read a paper about this also. I've said in the past that most of what I'm telling you in this class is true. I wish I could tell you, and then just 50% is maybe not true. I wish I could tell you which is that 50%. I think we're going to do better in this class because we really stick to the basic principles of cell biology. We're not so much on the cutting edge, but from time to time I'd like to take you a little bit to the cutting edge and show you where I think in the future interesting new discoveries could be. Okay, now, before we go there, now what I really hope to achieve in this class is I hope I can let you see in some new way, or see better. And well, let me try to illustrate that idea. So I'm going to warn you, I'm going to put you in the dark. In somewhat dark, because I'm afraid if I touch off, then I have to look with. So put us in low light, and we'll just imagine it's pretty dark. Okay. Now I have an object, and I drop the object somewhere on the floor. The object is somewhere on the floor. Let's say the object is important to me. It could have been a key to my home or my car. So now I'm in trouble. It's dark. I cannot really see. There are now two basic approaches for me, I believe, by which I can find my important object on the floor. One is I can go on all fours, I will do it, and just feel around everywhere on the floor until I've covered everything here around the podium. So go over, and I can't see, right? And just feel for it until I've found my object. It will work. You see this advantage. It will work, but there's a better way. The better way would be to increase the light in the room to maybe like, I'm not sure if that was the light I had before the presentation. So if I increase the light in the room, I can see my object much better. But this is really something I hope to achieve in this class. And if you look at this material in cell biology,